I want to welcome everybody to this third lecture of the Lefebvre Winter Series on Aging. This is a lecture series that honors the memory of Dr. Edward James Lefebvre. And he was a Galveston physician with a passion for geriatric medicine. Today, I have the great pleasure of introducing Dr. James Kirkland, who's a world-renowned authority in aging research and geriatrics. Dr. Kirkland is a professor of medicine and a director director of the Robert and Arlene Kogut Center on Aging at Mayo Clinic in Rochester, Minnesota. Dr. Kirkland is also a professor in the departments of physiology and biomedical engineering at the Mayo Clinic School of Medicine and holds an appointment as a professor of aging at the Nobel Foundation in the Netherlands. And he's also a geriatric doctor during his various activities. So Dr. Kirkland has a medical degree from University of Toronto, where he also completed his residency in internal medicine and a PhD. He also has a master's degree from the University of Manchester in England. He's board certified in internal medicine, endocrinology, and geriatrics. He started his academic career at the University of Toronto, where he reached the rank of associate professor, and then he moved to the Boston University in Boston, obviously, and stayed there for several years. In 2007, finally went to Mayo Clinic, where he's been ever since. He's been focusing his research activities on understanding the basic mechanisms of aging and also looking for innovative treatments to counteract these mechanisms of aging. In these treatments, it's now looking at translating them rapidly into patients to prevent the chronic diseases associated with aging and improve health in older people. So he's what we now call a true translational scientist. His research has been funded by a number of federal grants. He's actually the head of the Geroscience Network and several other grants, and has published more than 100 papers and several book chapters, and he's sat in a number of very important review panels and advisory panels at the federal level and internationally. So he's won also several awards, and so please join me in welcoming Dr. James Kirkland.
Thank you very much, and thanks for inviting me. And let me know if I'm going too fast, too slowly, boring you, whatever. And what I'd like to speak to you about is the possibility that we might be able to translate some interventions that affect fundamental aging processes into clinical application. Obviously, one of the things we can't do is study lifespan in humans, and the goal, I think, of most basic aging research is not to increase lifespan at all costs so that when you're 130, you feel like you're 130. Rather, the goal is to try to develop interventions that can slow down or the onset of age-related disabilities and increase the period of life of health span, the period when people are independent, free of pain, free of chronic disease, free of disability, as a proportion of total lifespan. So is there a way that we can target multiple age-related diseases by hitting the main risk factor for most of these diseases, which is aging itself? So this is a slide, sorry, I've got some asthma, that I stole from Felipe Sierra. He's at the NIH. And there's a hypothesis that Gordon Lithgow at the Buck Institute came up with a number of years back called the geroscience hypothesis. And that's that since aging, chronological age is the biggest risk factor, as it were, for the major chronic diseases like cancers or osteoporosis or heart attacks or strokes or Alzheimer's disease or diabetes, then is it possible that if we target fundamental aging mechanisms, we might be able to hit these diseases, which account for the bulk of morbidity, mortality, and health costs, as a group instead of targeting them one at a time. A lot more work needs to be done to test that hypothesis and see if it's really true. But it is remarkable that when you look at the bulk of the conditions that are most expensive and cause the most distress to people, they are these age-related chronic diseases in both developing and developed countries, and that aging is by far and away the biggest risk factor for all of them. So what is it that ties these diseases, the list of things I mentioned, like cancers or Alzheimer's disease or Parkinson's or heart attacks, etc., to chronological aging, to natural aging processes? And at the tissue level, there are at least 4 things that go on that seem to be shared in common between many of these age-related diseases and fundamental aging processes themselves. And those include inflammation of tissues, and this is non-microbial, what we call sterile inflammation, not caused by bacteria or viruses as far as we know, although that could be wrong. It tends to be low-grade, smoldering inflammation. You find inflammation also underlies what happens at the sites of atherosclerotic plaques, that predispose to heart attacks or strokes. You find this kind of inflammation surrounds regions of the brain affected by Alzheimer's disease. You find this kind of inflammation occurs in fat tissue and muscle in the case of diabetes and so forth. The second process is the one that I'll talk the most about. It's called cellular senescence, and I'll explain what that is in a moment. The third group of processes, which tend to tie age-related chronic diseases to fundamental aging itself, is what we call macromolecular dysfunction. So dysfunction of large molecules, DNA, proteins, including accumulation of aggregated proteins, including decreased turnover of proteins through a process called autophagy. Problems with what we call advanced glycation end products. So these are problems with complex sugars and how reducing sugars, smaller sugars, interact with proteins and DNA and cause dysfunction. And then a process called lipotoxicity. As you get past the 70s and 80s, you actually start losing subcutaneous fat. You start getting redistribution of fat. You get failure to store fat properly in fat tissue, and it starts being stored in places like the liver, in muscle, in bone marrow, and elsewhere, where fat molecules, especially fatty acids, are highly reactive and cause all kinds of macromolecular dysfunction. And so this loss of subcutaneous fat that people get that starts peripherally and works centrally, so you get thinning over the backs of the hands and spindly legs, that fat instead gets stored in places it shouldn't be and causes dysfunction. That's lipotoxicity, another form of macromolecular dysfunction. So the main large molecules, DNA, proteins, sugars, and lipids, get screwed up with increasing age, and you find this kind of messing up of these macromolecules at the sites of age-related chronic disease pathology. Again, you find this in Alzheimer's disease, you find protein aggregation, and in some of the frontocortical dementias, you find it with Parkinson's, you find protein aggregates in other conditions like in atherosclerotic plaques and elsewhere that we didn't know they were there before. And then the final process, which links fundamental aging mechanisms with age-related chronic diseases, is dysfunction of the stem and progenitor cells that are present in most tissues and give rise to new complex cells that drive the function of those tissues. So these 4 processes, inflammation, cellular senescence, macromolecular dysfunction, and stem and progenitor cell dysfunction, are all interlinked. One can cause the others to occur or get worse. And they're present in many tissues with natural aging, and they're present at the sites of pathology in many chronic diseases, even in diseases in children that are associated with fundamental aging processes, and I'll come back to that in a moment. So the biology of aging field has progressed from a long period of descriptive research where we were figuring out what aging looks like, what happens in various tissues with aging, through a period of hypothesis-driven mechanistic research, which is extremely important and must continue for us to maintain a pipeline of interventions, to a period of developing interventions. So as of about 2009, we started developing drugs that actually affect lifespan and the onset of age-related diseases in lower mammals, in mice. The first papers began to come out then. And as I'll show you in a minute, there are at least 25 drugs now which will do this in mice. We're approaching the period where we're thinking about translating these drugs into people and how we do this. And what's going to be the best approach for translating these drugs into people? Where will they see their first use? And then eventually application. So we're not at the translation point yet, although there are a couple of studies that have one has just been published, which sort of is beginning to bridge that gap. We've got a long way to go. So I'm not going to go through all of the drugs, but there's a fairly lengthy list now of drugs that have been published that affect either lifespan or health span in mice. The first one was to be found was rapamycin. But we've known about other interventions, like caloric restriction, for over 100 years. And it seems that many of the single gene mutations that extend lifespan in various species and caloric restriction share a lot of properties. So many of this, at least 2/3 of the single gene mutations that extend lifespan across species are on pathways that resemble those that caloric restriction targets. Of course, caloric restriction is not going to be, 30 or 40% caloric restriction throughout life is not something we're going to be able to translate easily to humans, especially in the face of an obesity epidemic. But what we have learned in experimental animal studies is what some of the molecules are that are activated in caloric restriction that tend to delay onset of age-related diseases and age-related dysfunction. So caloric restriction would have a lot of side effects. It would be difficult to introduce. People who are calorically restricted get tired, they get cold, they take to bed, they don't have sex, all kinds of things go wrong. They may live longer, but who wants that? The idea is to use this as a paradigm to figure out what are the pathways that are affected. Can we go downstream? Can we do what caloric restriction does with respect to life and health span and onset of age-related diseases without actually calorically restricting people? And rapamycin, this first drug that was found to affect fundamental aging processes, lies clearly on that pathway. It's heavily involved in pathways that mediate protein production turnover, as well as inflammation, cellular senescence, and stem and progenitor cell dysfunction. Since then, out of something called the Interventions Testing Program, which is run by the National Institute on Aging, and other studies, a number of other drugs have been described which affect life and health span. Actually, before rapamycin was even discovered, there were some papers that didn't really get the recognition they should have, that showed that angiotensin-converting enzyme inhibitors can extend lifespan in rodents. So the first papers came out of Argentina. They weren't really noticed in the early 90s, but they preceded the rapamycin study. And then it's been found that alpha-estradiol, 17-alpha-estradiol, which is an estrogen-like compound, in fact, it's the same as beta-estradiol that causes feminization in women, extends lifespan in rodents, especially males. Alpha-estradiol is not feminizing, and it does not act through estrogen receptors. So it's doing something which is different from beta-estradiol. It extends lifespan in male mice at the right doses by as much or if not more than rapamycin, and we don't completely understand yet how it works. But this is a readily translatable thing because this is circulating in all of us, including males. So it exists in things like Premarin and some of the older estrogen compounds, so we know that it's safe. And this is going to be low-hanging fruit for some drug trials. Metformin has been found to increase lifespan in rodents. It also delays onset of age-related cancers, possibly dementias, and certainly atherosclerosis and metabolic syndrome in humans. Furthermore, there's a study indicating that metformin, which is used for treating diabetes, results in improved survival in diabetics on metformin over non-diabetic control individuals who don't have disease, who are in their 70s. So there was a trial of almost 500,000 people where they found that in people at 75, if they were on metformin, their survival was greater without diabetes than in people with, the survival of people with diabetes on metformin was greater than the survival of people who didn't have diabetes and weren't on metformin. So it is really pretty fascinating. And we've confirmed this within our own 7 million medical records at Mayo. There are a bunch of other drugs that I've listed that show some promise, including acarbose and others. And I'll come back and talk about senolytic drugs in a moment. It's our main area of interest. So we've been interested in fat tissue for some time. Fat tissue is at the nexus of a lot of processes that are related to aging and age-related chronic diseases. I mentioned caloric restriction before, can delay some age-related chronic diseases as well as extend lifespan across a variety of species, maybe not higher primates, but it certainly extends health span in higher primates. And it acts mainly through fat tissue in that context. Obesity is the opposite effect. Obesity is associated with accelerated onset of Alzheimer's disease, non-hormonal cancers, atherosclerosis, and all kinds of other things that are characteristic of an accelerated aging-like state. If you remove certain kinds of fat tissue surgically in mice, you greatly increase lifespan. And it's been found that fat tissue is the main source of inflammatory cytokines that accompany frailty in older rodents. So age-related muscle loss and frailty and dysfunction in rodents, the main source of the circulating factors that are related to this appears to be fat tissue. So we've been interested in studying fat tissue. Also, fat tends to be the largest organ in most people. A woman with a body mass index above 35, for example, is more than half fat tissue. A male, it's something like a BMI of 40. So we have a lot of fat tissue. Fat tissue is very rich in progenitor cells that can give rise to new fat cells. Up to 50% of the cells in fat tissue are progenitors, anywhere from 15 to 50%. And these cells can become senescent. And we find exuberant senescence in fat tissue with aging, in diabetes, and in obesity in certain other states. So there are multipotent progenitors that can become pre-adipocyte Caesar cells that can become fat cells, and they can be pushed into becoming fat cells through a process called differentiation. Or they can be gotten rid of through programmed cell death called apoptosis or cellular senescence. And I'm going to focus on cellular senescence for a minute, because we see one of the biggest tissues, as I mentioned, where we see senescent cells accumulating is in fat tissue. So what are senescent cells? Senescent cells were first discovered by Moorhead and Hayflick back in the late 1950s. And they found that if you put human cells into tissue culture, they can only divide a certain number of times, and then they stop dividing even if growth conditions are optimal. They don't die. In fact, they'll live in an incubator for 10 or more years. In fact, they're resistant to death. You can drop serum, and they are very resistant to dying through the process of apoptosis, much more resistant than normal cells. They become big. They develop a large nucleus, this area, a single nucleolus. They're 3 times bigger than a normal cell. They contain more protein. They produce more protein. And they with a particular enzymatic reaction, you can that's moderately good but not perfect for identifying these cells. They'll stain blue because they've got a particular enzyme called senescence-associated beta-galactosidase. And for those who are scientists in the audience, this beta-galactosidase reacts at a different pH than regular lysosomal beta-galactosidase. So you can spot these cells. They also have DNA damage responses going on. So this is the nucleus. And there are multiple spots where there's phosphorylated histone that indicates that DNA is undergoing damage responses. And we also know that DNA gets folded up in these cells and forms something called heterochromatin. So these are human preadipocytes that are senescent. They've been passaged in culture for a number of times. Now, there are about 30 or more processes, 30 or more factors, stimuli that can cause a cell to become senescent. A cell integrates these signals and decides whether to become senescent or not. Senescence is a process very much like differentiation, when a non-specialized cell becomes a specialized cell. So what we call transcription factor cascades get activated. Around 2,500 genes change in their expression. DNA gets folded, and over a period of anywhere from 10 to 25 days, depending on the cell type, the cell becomes senescent. It becomes resistant to removal by apoptosis. It produces inflammatory factors, that inflammatory cytokines. They also produce things which attract immune cells in and around them. And they produce things which degrade and alter the matrix in which cells reside. So even though there are very few senescent cells that accumulate even with aging, because they produce a lot of inflammatory cytokines, chemokines, proteases, other things, highly reactive molecules, at least 450 of them, proteins and peptides alone, a very small number of these cells can have a very big effect, as I'll show you in a moment. So we find that with aging, this is what happens in rats. A 3-month-old rat's equivalent to around a 10-year-old human. 24-month-old rat is like a 80-year-old human. We find senescent cells accumulate here. They're stained with that enzyme I was telling you about before. You can see they follow a linear pattern. They follow where blood vessels are. That's because preadipocytes and endothelial cells, the two main cell types that become senescent in fat tissue, follow blood vessels. With obesity, the same thing happens. So this is a lean rat. This is a rat that's been fed what we call a McDonald's diet, so it's become obese. And again, you can see the senescent cells are following a blood vessel. That's what happens in people. So this is 37-year-old woman, 30-year-old woman. This 37-year-old woman is lean. This 30-year-old woman is, moderately, just mildly obese, arguably. And you can see the senescent cells are following a blood vessel in this person, despite the fact they're younger. So there's something about obesity and the high sugar that occurs in obesity, some of the lipids that occur in obesity, the cell proliferation that occurs, that can drive cells into a senescent state, as well as with aging and other conditions. So amongst the cell types that I showed you before, there are many cell types in fat tissue, but amongst the cell types are those preadipocytes I was talking about before that can turn into fat cells. With increasing age, you find increasing abundance of senescent preadipocytes. This is using methods where we take cells from animals, in this case, and make sure we've got highly pure populations of preadipocytes. So we know that both endothelial cells using similar sort of approaches and preadipocytes can become senescent. Same thing happens in people. You can see that's senescent cell from this 69-year-old individual. You don't see them in the younger individual unless they have diabetes or have been radiated or had chemotherapy or something. If we take senescent cells in culture and take the culture medium from them and put them onto normal fat tissue, we provoke inflammation of that fat tissue. So even a very few senescent cells produce enough factors into medium in culture that if we take that medium in the black from senescent cells compared to medium taken from non-senescent cells, we induce all these inflammatory cytokines, chemokines that attract immune cells, and then things like hemostatic factors that cause blood clotting and all kinds of other problems. If we take preadipocytes from lean younger individuals and we look at the amount of something called MCP1, which attracts immune cells, we find it's lower than if we take conditioned medium from preadipocytes from obese, age-matched individuals. There's twice as much MCP1 being produced. And there's much more migration of macrophages, these immune cells, across a membrane that we see compared to a lean younger individual. So this is an obese younger individual. If we take the cells from the lean subject and put them into culture the way Hayflick and Moorhead did and make them divide multiple times, we find more MCP1 production. If we take cells from a lean older subject compared to a body mass index matched lean younger subject, the cells from the older subject in culture, primary cultures, produce more of this MCP1 and cause more macrophages to migrate. So there are things that these senescent preadipocytes are making that cause these immune cells to migrate into fat tissue, and these cells migrating into fat tissue have been associated with inflammation, with diabetes, and other things that fat tissue, when it goes wrong, can cause. With increasing age, these the preadipocytes that we have lose their ability to turn into fat cells properly. And preadipocytes don't have insulin receptors. As they turn into fat cells, though, preadipocytes acquire insulin receptors and insulin responsiveness. So if you don't have preadipocytes that are turning into fat cells properly, one of the consequences long-term can be impairments in insulin responsiveness and ultimately diabetes. So with increasing age, just natural aging, the ability, shows what happens in a rat, a young rat, about a 60-year-old rat, an 80-year-old rat. The ability of preadipocytes to turn into fat cells grown under identical conditions in parallel from a young compared to a middle-aged compared to old animal declines. So there's less, this has a lot of lipid and these are beginning to turn into fat cells, accumulating fat. These don't do as well. And the same thing happens in people. Now, if you take conditioned medium from senescent cells and you put them onto young differentiating preadipocytes, you abrogate, you prevent the cells from turning into fat cells properly. Where if you take conditioned medium from non-senescent cells and put them onto differentiating fat cells, they acquire lipid properly. This is just, I'm just going to go through this briefly. I don't want to scare non-scientists in the audience very much, but two of the main transcription factors, these are things, sort of the conductors of the orchestra that allow an undifferentiated cell to turn into a differentiated cell. The two main transcriptional regulators that turn preadipocytes into fat cells are this guy, CBP alpha, and this one, PPAR gamma. And incidentally, this is a target of a class of drugs that are used for treating diabetes. The ability of these things to turn on decreases with increasing age. So this is from old animals compared to young animals. The ability of CBP alpha to turn on declines and PPAR gamma to turn on declines. Both of these things have to turn on properly and stay on for a fat cell to be insulin responsive. If these things are low, you become diabetic. When we take conditioned medium from these senescent cells and put them onto cultures, from young individuals, we recreate what happens with natural aging in the previous slide, where these things go down with natural aging. If we take senescent conditioned medium from senescent cells that's been exposed to senescent cells and expose the non-senescent cells to this stuff while they're differentiating, we turn down expression of PPAR gamma compared to medium taken from non-senescent cells. And the same with CBP alpha. And this translates into poor insulin responsiveness in tissue. So if we take conditioned medium from senescent cells, the culture medium senescent cells have been growing in, and we apply it to fat tissue, we prevent insulin from turning on its downstream targets in that fat tissue. So there are things senescent cells are producing that come from fat tissue, if the senescent cells are from fat tissue, that prevent fat tissue from responding to insulin. They cause what we call insulin resistance, and this predisposes to diabetes. Now, we know that, we found that, I'm not going to go into great detail, but we found a series of, there's some signaling molecules called JAKs, which mediate the effects of inflammatory cytokines, these things that senescent cells produce, on tissue function. And one of, amongst the main cytokines are ones that signal through this particular pathway. We find that these things are upregulated in fat tissue with increasing age. So I don't have time to go into the details of exactly why we did it, but because of this and because we know that some of the inflammatory cytokines that we believe impair insulin responsiveness of fat tissue signal through this molecule, we added drugs that inhibit JAKs 1 and 2 to human preadipocytes that were senescent. And we found that we were able to drop down the amount of production of the inflammatory cytokines that cause some of the dysfunction with, that's provoked by these cells, and some of the chemokines that attract immune cells, as well as hemostatic factors and other things. We found that if we take cultures from old animals that are producing these inflammatory cytokines, and if we treat them, they're producing a lot of these things, like interleukin-6 or this MCP1 I showed you before that attracts macrophages. or these MMPs which degrade collagen and the tissues that surround cells. We find a lot of these things are produced by cultures of preadipocytes from old animals that contain senescent cells. When we add these JAK inhibitors, we lower expression of these things. When we add these JAK inhibitors to cultures from old animals or people, I showed you before that cultures from old animals or people, this shows what happens in rats, exposed to differentiation-inducing medium, don't accumulate lipid. But when we add these JAK inhibitors to these cultures, we restore ability of these cells to accumulate lipid. And we restore expression of those transcription factors, PPAR gamma and CBP alpha that I showed you earlier. So by adding these inhibitors, we're reverting what happens with respect to these transcriptional regulators, lipid accumulation, and so forth, in aging animals with senescent cells back to what happens in younger individuals. So we gave these kinds of drugs to mice and rats. This shows mice. And I mentioned before that you lose subcutaneous fat. Well, so do rodents when they get old. And they develop this lipotoxicity that I talked about before. We were able to get them to restore their fat tissue when we gave it to very, very old animals. So these are equivalent to almost 100-year-old mice. And we were able to get them to restore their fat tissue. We were also able to get them to animals that were on a high-fat diet, that McDonald's diet that I talked about before that causes diabetes. We were able to restore glucose and insulin responsiveness in those animals by treating the animals with these specific JAK1/2 inhibitors. So this shows something called a glucose tolerance test and an insulin tolerance test, and we improved these in high-fat fed animals that were developing diabetes. We also improve frailty in these animals, things like their aerobic capacity, their metabolic rate, and their activity in cages. So these are very old animals, and we were able to restore activity in them. So this led us to the notion that maybe we should get rid of senescent cells altogether. So what we decided to do was to take something called a senescence activated promoter, in this case P16, which is a thing which turns on genes and that is activated by senescence. And we took a particular fragment of that promoter that we know is very important to senescence in cultured cells. So we didn't take the whole promoter region, all this DNA. We took a specific region of it that we knew is activated with senescence. And then we attached to it something called ATAC, which is a construct. It's called apoptosis through targeted activation of caspase. But basically, what it has, it's a fusion of several proteins, and it includes a protein that will cause suicide of a cell. It'll cause a cell to die if you bring two of these proteins together. So these proteins are made as single proteins that are not brought together. But if you put in a drug, AP20187, which doesn't do anything to normal cells, it'll cause this region of one of these fusion proteins that contains this killer molecule to become adjacent to another, which activates the suicide gene. So our theory was that if you put this, if you made mice that had this kind of construct in there, and then if you gave this drug, you could selectively eliminate senescent cells and only senescent cells. So we then worked with Jan van Dersen and others to actually make the mice. He felt we were torn between the P16 and the P50, some other promoters, but we eventually settled on the P16 promoter. We added, Tamara's idea was to add a GFP to this, so we could track these cells so they'd be fluorescent. And we decided to breed these mice onto either an accelerated aging background or high-fat feeding, and we've done both, and I'll show you both. And we found that indeed, in fat tissue, this is inguinal fat tissue down in this region in mice, that in mice that are not given the drug, you can see these green cells. When we give the drug, we eliminate the green cells. When we look for this senescence-associated beta-galactosidase, that bluish stain I showed you before in senescent cells, this is mice that are not given the drug. This is genetically identical mice that are given the drug. So we're able to eliminate the senescent cells. And furthermore, with that fluorescent tag that we put in there, we're able to sort out the senescent versus the non-senescent cells, and figure out what's in them and what makes them tick. So we found the main source in fat tissue in these animals with advancing age of inflammatory cytokines and PAI1 and other bad things is a senescent cell fraction and not normal cells in their fat tissue. And it's specifically in what we call the stromal vascular fraction where preadipocytes are and endothelial cells are, not the fat cells themselves, and it's specifically in the senescent preadipocytes and endothelial cells. So this is a mouse that was not given the drug to get rid of senescent cells for a number of months. This is an identical mouse, litter mate, a brother of this one, genetically identical, that was given the drug. So we had an obvious effect on the phenotype of these animals by clearing their senescent cells. We only cleared 30% of their senescent cells, and their senescent cells only accounted for around 3% of all of their cells. So there's a tiny fraction of cells that we're killing. We were able to restore their subcutaneous fat in much the same way as those JAK inhibitors that I talked about before. And we were able to restore their glucose when we used this kind of method in diet-induced obesity, so that these animals were diabetic. This shows the high-fat fed control animals, and this shows what happens when we give drugs to clear senescent cells every few weeks. So we get a restoration of glucose responsiveness. We've now developed drugs that specifically kill senescent cells and not normal cells, and we get the same thing. So we call them senolytic drugs. We phenocopy what happens in these animals. So there's emerging evidence now, and I don't have to, I've only talked about fat tissue, I've only talked about diabetes, but we've done, and other labs in our group have done equivalent kinds of work with a variety of other conditions and find similar sorts of results, both with the genetic models and with the drugs. So we're finding, as I showed you here, we're able to have an effect on diabetes associated with obesity, on age-related lipodystrophy. We're now finding that we're able to enhance cardiac function, heart function in these animals, and we're, conversely, when we transplant senescent cells into mice or rats, even very small numbers, we're able to cause diabetes that will last for up to 6 months, just with a single transplant of 2 million cells in an animal that has 500 billion cells. We're able to cause cardiac dysfunction, and these cells are only in one place in the animal. They don't migrate out of that place. So despite the fact they're nowhere near the heart, we're getting decreases in their ejection fraction and their fractional shortening by echocardiography. And we're getting all kinds of, we're getting frailty phenotypes and all kinds of things, even with transplanting small numbers of senescent cells. So now we've made senescent cells we can transplant that we can kill by putting in gene constructs where we can selectively kill them, and we're also treating the animals with senolytic drugs to see if we can get similar sorts of effects. We don't know that yet. We're finding that if we clear senescent cells from animals that have genetic mutations in them, that when they're given a high-fat diet, they'll develop plaques, atherosclerotic plaques in their blood vessels, that we're able to prevent development of those plaques. I've already shown you effects of frailty and on frailty and sarcopenia in both the genetically cleared animals and animals that have been given JAK inhibitors, but we don't know yet, but we're in the process of testing this with senolytic agents. We know now that we're able to prevent a lot of side effects from chemotherapy. Part of the way some forms of chemotherapy work is to cause senescence in cancer cells. We find that we're able, if we, after the chemotherapy, if we give senolytic drugs, we're able to increase the amount of chemotherapy we can give without causing toxicity, and we're able to get more efficient clearance of cancer cells because some of the cancer cells have been put into senescence, but they're not necessarily killed. If on top of that we kill the senescent cells, we're getting better outcomes with respect to cancer. With radiation, it's quite interesting. So we have animals where we radiate one leg, and then we find that after a couple of months, they have difficulty walking on a treadmill. Their treadmill endurance goes down. When we clear senescent cells with one treatment. We give these drugs orally for 1 day. We're able to get improvement in exercise endurance that seems to last six to 7 months. Collaborators of ours are working on cancer, and things look interesting, and then all of these other conditions. So it's beginning to look promising, but there are positive effects of senescent cells too. You don't want to, if you get rid of them, you interfere with wound healing and a bunch of other things. But the nice thing is that with senolytic agents, you just have to treat an individual for a day, clear the things. You won't get resistance because the cells don't divide. It's not like, cancers or microbial infections where cells keep dividing and eventually get resistance to the drugs. You kill them, you can give the drugs to a person for 1 day and then in an animal, not again for a very long time. So side effects are not a real concern because the drug is long gone. And so it might be a promising kind of treatment, but we're looking really hard for side effects. If you continuously clear these cells, we do know that you get problems with wound healing and tissue regeneration and other things. So we wouldn't want to give them continuously. So the steps that we've got underway are needed for, and I've only talked about senolytic drugs. Other groups and some people at our place are also working on all of these other compounds. A lot of people here are working on rapamycin, for example. And then there are various groups around the world working on metformin, on alpha-estradiol, and acarbose, and all these other kinds of approaches that might be complementary and add to what we see with senolytic drugs, or our senolytic drugs might be complementary to those drugs. So there's a whole range of approaches now for targeting fundamental aging mechanisms. And I think people are beginning to see similar sorts of things with other kinds of treatment. I've only focused here on senolytic agents, but there's some hope that we might be able to see this, at least in mice, with other kinds of drugs. The real question is, can we translate this into something we can do for people? And how do we do that translation? So one of the things that is a barrier is what are the right mouse models of human chronic age-related diseases? Many of these diseases are uniquely human, and there are no very good models. For example, Alzheimer's disease is almost uniquely human. Dogs get something like it. It looks like dolphins get something like it, but nobody's even sure in those particular situations how similar it is. Most models have been made of Alzheimer's disease where you interfere with a single gene and you get Alzheimer's-like states, but you get them occurring in 4 or 5 week old animals, which is like 10-year-old people. And there have been about 300 drugs so far, according to a review a few years back. It's not my area, but according to this review, that have been developed in Alzheimer's disease mouse models that have not worked in people. Because in people we're dealing with a disease that involves multiple genes, is highly complex, involves all kinds of environmental exposures, and occurs in the context of aging, where the mouse models are getting this disease at the equivalent of 10 years of age. So, and the same is true of a lot of other human diseases that appear to be, relatively uniquely human. The nice thing about diet-induced obesity and diabetes is that occurs across species. So you can feed a high-fat diet, especially to some forms of rats, and they'll get diabetic. Some mice are resistant, but like with people, there are some rats and other models where you can get pretty good diabetes going, giving the same kind of intervention as you'd use in people. And those kinds of animal models, I submit, are reasonable. The other kinds of animal models that are reasonable are prodroid models. That is these kids who get accelerated aging syndromes. Those are single gene traits. And there are some pretty good mouse models for some of those. So they'll be useful for developing intervention studies. And then things like the effects of radiation or chemotherapy are pretty translatable because many of the same processes seem to occur in rats, mice, dogs, or monkeys in response to chemotherapy or radiation as occur in people. We need to think hard about beneficial function of cellular senescence in the case of senolytic drugs. But with all these other drugs that target different kinds of pathways, we have to think about beneficial functions of those drugs. For example, rapamycin, adverse effects of those drugs. For example, rapamycin might conceivably have a lot of side effects, especially if it's given continuously in high dose. Even metformin, which is a relatively safe drug, might. But we need to think about what are the side effects, and especially long-term side effects. And these are very hard to predict in people based on what you see in animals. And so we need to have a real sense of caution as we develop these agents. And it also means that we can't be giving these kinds of agents to regular healthy people under most circumstances. We've got to be thinking about giving them to people who've already got manifestation of a disease, and especially if there's going to be any risk of side effects, to people who would be quite sick if they didn't have some kind of treatment for those diseases, because we're always talking about risk-benefit ratios. And so we can't be giving drugs to 20-year-old people in the expectation it's going to delay age-related dysfunction when they're 80. First of all, it would be hard to study, and second of all, any side effect at all would disqualify that drug. And there being no patent protection, patents last 18 years or whatever. We need to think about various kinds of biomarkers, especially of mechanism and pharmacodynamics. We don't need mechanism biomarkers for clinical trials, but you do need them for adjusting dosing and ensuring compliance. For senescence, we are working on trying to develop blood and tissue biomarkers that we can use to see if these drugs are actually clearing senescent cells, as well as imaging methods. And then we need to think of clinical trials approaches. Where would you use these drugs in people in a rational sort of way? So the kinds of animal models we can think of are old animals, frail animals, old animals with geriatric syndromes, things like dogs with incontinence, for example, or hamsters with osteoarthritis. We need to be innovative and think out of the box a little bit. As I mentioned before, we need to think about using animals that are exposed to clinically relevant insults to test resilience. So if we do surgery on the animal, if we give it chemotherapy, radiation, bone marrow transplant or something, are we able to reduce adverse effects from those things in older animals that get these kinds of things that we do to patients all the time? Will these drugs protect against those things? We need better animal models for testing mechanism, although there's some interesting things. You might have read about the parabiosis models where you join a young and old animal together, and that's going to be a very interesting model for developing some of these drugs where we can look at interaction of circulating factors. Cell culture models are getting better and better, and there may be situations like with Alzheimer's disease where at least screening will be better in human cell culture models potentially than the animal models. That was just recently published by Rudy Tanzi out of Harvard. And then we, especially for drugs that affect cellular senescence, we need to have strategies to distinguish, to know if we're dealing with cause and effect. In other words, if we have a senolytic drug, is it really working on whatever improvement we see by killing senescent cells, or is the drug doing something else to other kinds of cells through a completely different mechanism? It has nothing to do with senescence that is causing the benefit. And we just see senescent cells occurring as an epiphenomenon, something that occurs but is not related to the actual treatment. So these things all have to be thought about and developed further. So some clinical trials considerations we need to think about, not only with senolytics, but any drug that targets fundamental aging processes with the expectation we might be able to target age-related chronic diseases as a group, is that we need to think of measurable, clinically relevant outcomes for clinical trials that are appropriate to older populations. And we need to develop animal models that reflect this so we can test these things in animals before we go to people. And there's been remarkably little thought about doing this in our field because there have been remarkably few interventions so far, and now we've got them. We've got to catch up to other fields and think, what are the clinical trials paradigms going to be? How will we do a clinical trial? And then how do we develop an animal model that is going to reflect that trial so we can do that first before we do anything to people? We need to think a lot more about biomarkers, endpoint biomarkers of longevity. We're not going to develop in any near-term kind of work. We're not going to be able to get the FDA to accept the way they do blood pressure as an endpoint for a clinical trial to reduce stroke risk. We're not going to be able to come up with a biomarker where we can do that as an endpoint for longevity. That would take 100 years to do the kinds of trials you'd need to do to show that an endpoint reflects longevity reliably enough. that the FDA would accept it, because you'd also have to show that modulating that biomarker or a modulation in that biomarker by a drug would be associated with increased longevity, because that's what people had to do with blood pressure before it was accepted, or blood glucose, or any of these things. This is going to be very hard to achieve, and probably isn't a kind of thing that we can do. But other biomarkers are going to be important. That's developing biomarkers to test whether the mechanism we think we're targeting is really being targeted. As I mentioned before, you don't need that for drug development formally through the FDA, but it's a good idea to have it. And then we do need dosing biomarkers. Like, is the drug hitting the target it's supposed to? If you have to make sure people are compliant, you have to make sure you're giving the right dose of the drug. This is actually a lot of work. We need to think of situations where we can give these drugs to people who already have symptoms or are imminently at risk of developing symptoms. As I mentioned before, we're not going to be able to give these kinds of drugs to healthy 20-year-olds in any in any sort of time frame soon. We're going to have to think of where you can do clinical trials that last a few weeks, months, or a couple of years. We're going to have to use these things where there's a compelling clinical need, if there's any risk of side effects, or if it's an investigational new drug that doesn't have a track record. Repurposed drugs, like metformin and things, we might be able to get away with using these kinds of agents in trials where there's less of a compelling clinical need, because we know the side effect profile is very good. So we have to match the side effect profile with the clinical need. We have to think about recruitment in some of these situations and other considerations. So where are the places where it's likely that we're going to use things like drugs related to rapamycin, or things related to alpha estradiol, or things related to metformin, or senolytic drugs, or drugs that inhibit the secretory phenotype of senescent cells, or any of the other 25 drugs that are coming along? And I can think of, and our group, the Stereoscience group that we were talking about earlier, has come up with maybe 6 areas, but this is a moving target and we're only beginning to think about this. One of the areas that we're beginning to think about is simultaneous alleviation of multiple comorbidities, because older individuals tend to have tend to be either very, very healthy, or have a whole lot of medical conditions and be on a whole lot of drugs that all occur at once. And there's epidemiologic evidence that if you get one age-related chronic disease, the second one is not going to be far behind it, on average about 3.6 years. So these conditions cluster in people at a particular stage in their life, and you've probably seen that, where people just sort of fall apart, and they wind up with 5 different problems all at once, and they're on 10 or 15 drugs. So if you hit one of these conditions with one of these drugs, do you improve the others? So that's one kind of clinical trial paradigm. Another would be is if a person comes down with one of these age-related chronic diseases, can you delay the time to the second one occurring? So there's active planning going on around the country for these kinds of trials, and a lot of discussion and protocol development. Another situation where these kinds of drugs could be used is where there's so-called accelerated aging, which is it's really hard to talk about accelerated aging. You can age a mouse by buying a sledgehammer and hitting it good and hard. That's not really aging. You're killing it early. And so accelerated aging is always a tough sell. It's much easier to sell prolongation of lifespan. That's a much harder bar to meet. But there are some conditions that we know are associated with fundamental aging mechanisms that result in accelerated aging-like states. One of the groups is childhood cancer survivors, so people who've been treated for leukemias or whatever as kids. They're dying in their 30s and early 40s, some of them, not all, but some of them, of accelerated aging-like states. So they're getting Alzheimer's disease, they're getting second cancers, they're getting frailty, they're getting diabetes and severe metabolic syndrome, and all the rest of it. So some of these people look like they're 60 or 70 when they're 35. And they're dying in their early 40s. And these are people, especially the ones after the early 90s, when we started adding high-dose radiation to chemotherapy for treatment of childhood leukemias. So the people, especially after 92, are beginning to get this syndrome. We also see it in bone marrow transplant survivors, where we haven't been able to use the subject's own stem cells to do the bone marrow transplantation. So people who are getting bone marrow transplant from other donors because of cancer. Some of these people, again, not all of them, Well, these people get a 10 times lethal dose of chemotherapy. So they have a gigantic whack of chemotherapy, and that's what kills the cancer in them. And it kills off their own bone marrow, and you have to replace their bone marrow. But they're getting a 10 times lethal dose. And this causes widespread cellular senescence, because that's how a lot of chemotherapy drugs work. They cause cellular senescence, especially in cancer cells. And we're beginning to find that these people, after 3 to 5 years, are getting an accelerated aging syndrome. So even if they survive their original cancer, they're getting second unrelated cancers. They're getting dementia. They're getting diabetes and metabolic syndrome, and they're getting frailty. So we view this as a very early group for trying some of these senolytic agents and other kinds of agents that target fundamental aging mechanisms. The prodroid syndromes, these are syndromes where kids have accelerated aging-like characteristics. There's very tremendous interest in the NIH for trying a group of these kids on some of these kinds of drugs. Diabetes due to obesity is attractive because, as I mentioned before, they get an accelerated aging-like state. And if we treat diabetes with these kinds of drugs, in addition to controlling the blood sugar, would we be able to control the complications of diabetes, like renal dysfunction, brain dysfunction, problems with heart attacks and strokes and other things that tend to go along with diabetes, and cancers and things like that. So these drugs may go beyond what insulin does, and they may slow down some of these things. And there's evidence now, as I was mentioning earlier, with metformin, which is used to treat diabetes, that this is in fact the case. Metformin hits something called AMP. It hits mitochondrial complex 1 and AMP kinase, for those in the audience who know about this, and ultimately affects things like mTOR and other kinds of processes that are related to caloric restriction things, these processes that slow down aging processes. HIV dementia is another situation where we see using these drugs early. Conditions associated with localized cellular senescence. This would apply to cellular senescence and things that modulate the secretory phenotype of senescent cells more than other kinds of things. So that would be osteoarthritis, because we can inject the drugs locally. Fracture non-union, which is associated with cellular senescence at the site of where the two pieces of bone that won't go together. Idiopathic pulmonary fibrosis, so we're developing drugs that we can put in aerosols for this condition. Otherwise fatal conditions, so idiopathic pulmonary fibrosis is in that group, primary biliary cirrhosis, certain cancers, HIV, dementia. These diseases seem particularly associated with cellular senescence and some other processes that are characteristic of fundamental chronological aging. Resilience I mentioned before, and that's do we improve responses to chemotherapy, radiation, elective surgery, rehabilitation after myocardial infarction. We have a trial going on now with drugs related to rapamycin in these people. At Mayo, we've got a trial underway right now. Immunization, this is the first published trial that came out. It came out on 24th of December, that a drug related to everolimus, which is in turn related to rapamycin, giving 1 to 3 doses of it 3 weeks before influenza immunization in elderly men resulted in tremendously improved immune responses and decreased influenza infection rates, as well as improvements in general immune function. They looked at every kind of immune function you can imagine. So they did this in 208 people in Australia, Novartis trial, just published in Science Translational Medicine. We're interested in recovery after pneumonia, and then intermediate outcomes like loss of independence in moderately frail subjects like Fried class 2 or 3 subjects. The geriatricians in the audience will know what that means. How long is it? Can we delay their first institutionalization with some of these agents? So I'll just end by saying I wouldn't give it a high chance, but there is a chance. I would have said it was 0 chance 10 years ago, a 1% chance 5 years ago, and maybe a 20 or 25% chance now, but not much more than that there could be a transformation in geriatrics coming along. And a lot of people are betting a lot of resources on this. So the drug industry and various groups are putting huge amounts of money into this because, as you can imagine, if any of these approaches work, it would truly transform medicine as we know it. Slim chance. I don't want to be over-optimistic. We're going to find all kinds of bumps on the road, all kinds of side effects, all kinds of problems. And these things always, no matter how optimistic people are, bad things happen as we try to develop these things. We have to be prepared for that. But currently, at least I do some geriatrics from time to time, and I'm getting a bit sick of prescribing better wheelchairs, better walkers, and better incontinence devices. I think it would be a lot better if we could delay that whole mess of things. And it may just be possible that we may not be far from doing that, maybe. So the hope, and this is an official hope of the EU, they've put this in their 2020 vision. You know, they're spending huge amounts of money on this in the European Union. It's right up there with climate change as one of their 3 priorities. Are we going to be able to delay onset of age-related diseases and improve health span? And can we do this based on recent advances in the basic biology of aging? Really a hypothesis at the moment, not reality yet, but there's a possibility. So in conclusion, there are exciting new agents that target fundamental aging mechanisms. They appear to ameliorate frailty and enhance lifespan and lifespan in mice, but in mice, we don't know with other species. And these could lead to maybe, possibly, clinical interventions in humans if preclinical studies demonstrate effectiveness, and especially low toxicity, which is something we need to focus on, and if we can establish the right clinical study approaches. I'll just conclude by thanking people in my laboratory, our many collaborators, and our funding institutions for helping us with this, especially Tamara Ciconia, who together with us came up with the idea of the IncaTac animal, and Nathan and Yan, who helped make those mice, and then the people who helped develop some of the senolytic drugs, and as I mentioned, our many collaborators and colleagues. Thank you very much.
Well, thank you very much for this fascinating and exciting talk. It's 6 o'clock, so we have time probably for one question, and then we can mingle with the wine and cheese session. So I need the takers for the question. Yes.
So, could you catalog cells kind of develop into crossing cells?
Any proliferating cell can by definition, because the definition of cellular senescence is loss of replicative capacity without becoming differentiated and without dying. It's a bad definition. The definition has to change. It looks like differentiated cells that are not capable of dividing are perfectly capable of developing a senescent-like state when you look at their expression profiles and what happens to their DNA. There is no good single unitary definition at the moment, but it does appear that no matter what the cause of induction of senescence, over time, most senescent cells exhibit very similar phenotypes, ultimately, if they're left long enough. And there's some work, not by us, but by others at another institution that is beginning to indicate that. We do know that in the short term, senescent cells, even from different fat depots, for example, are quite distinct in what they'll produce and what they won't. But it looks like if you wait long enough, they sort of turn into this terminally differentiated state, which is remarkably similar across cell types.
Thank you so much again. And
UTMB Health, working together to work wonders.